Continuing on with chapter 6, after covering sort of the introductory, let's get started part of this chapter, I want to get now into the details of what's happening at the retina itself. So we have this idea, of course, that light is going to enter into the eye. The lens will focus the light rays to the retina, the interior lining on the inside of the eye. Which is now lined with many neurons. There are other neurons besides photoreceptors, by the way. So that's our first point here. So the photoreceptors would be the beginning, the starting point of a long series of events here. Photo means light. So the idea, of course, is that these are essentially specialized neurons that will respond to incoming light. And then, of course, they're going to have an axon that will synapse onto some other neurons. And then this whole, all of this information that's encoded about the properties of the visual stimulus will then be organized, processed, and transmitted to the brain for additional processing. A couple of features that we want to know about the retina before we get into the details of what these receptors are like and what some of these other neurons are like and what they do. Is the fovea, right? So the idea is that when you look directly at something, we call that fixating or foveating. And the idea is that the image of that object, if you're looking, say, directly at your computer screen right now or a phone screen, that image is falling directly onto the center of your retina, and that is called the fovea. And also in particular, at the very center of the fovea is the even highly more specialized region called the macula. And this is where we find, and I mentioned this when we talked about the general differences between rods and cones before, I think, I'm keeping all my videos mixed up here, which is that we have a very high concentration of cells here, right? The idea is that whenever you need a lot of detail about some stimulus, you need more receptors to extract all of that detail, right? And it's in a way, it's kind of similar to your sense of touch. If you think about your fingertips, for example, you have on your fingertips all of these little creases and folds of your fingerprints. And one of the things that this does is it increases the surface area of the skin on your hand so that you can embed more touch receptors on your hand. And of course, when you touch something with your hand, you're going to get a lot more information and a lot more detail about it compared to if you're touching something, say, with your elbow or something, right? Because you just don't have nearly the high level of touch ability with other parts of your body compared to what you have with your hand. So that similarly, this is happening with your eyes, is that you have a lot of receptors that are packed in very closely and densely in the center of the retina. Whereas in the periphery of the retina, of course, you still have a lot of receptors out there because you do have peripheral vision. You can see in the sides of your field of view, but clearly the level of detail and clarity is not nearly the same as it is in the central vision. And that's because of the organization of the fovea and the way that the cells work in the fovea. Also, another important landmark on the retina is called the optic disc. And it's not really a disc so much as it kind of maybe looks like a disc when we look at certain photographs of a retina. But the idea is it's actually a gap. It's a hole in the back of the eye, because these photoreceptors, as I noted, have axons, and these will synapse onto the dendrites of some other neurons that are also in the eye. And ultimately, all the activity of all of these other neurons has to be transmitted to the brain. So what has to happen is that the axons of these neurons will bundle together to form a nerve, right? And that nerve is the optic nerve, and it leaves the eye and it goes to the brain. And so we're calling it a nerve, not a tract, right? Because it is part of the peripheral nervous system at this point until it hits the thalamus. But what's going to happen here is that we have to have a gap, a hole in the back of the eye to allow these axons to leave and go back to the brain. What's also convenient here is that this provides a nice access point for the blood supply as well. So we need blood vessels to provide nutrients to the neurons inside the eye. And so we use the same point to get that blood supply going in there as well. So this technically then is a blind spot, since there would be no photoreceptors located at this point. We don't typically notice the blind spot in our vision. And a couple of reasons for that. One is that on each eye, on each retina, the blind spot is in a slightly different position. So that means that when you're looking at something with two eyes, the blind spot of one eye is being compensated for by the other eye, which can see whatever object falls on that location in the visual field. But even if you closed one eye, you're still not likely to notice the blind spot because it's very small. It's potentially true that we also kind of adapt to that and compensate and fill in the gaps a little bit with higher level visual processing. But also importantly, our eyes are really always moving. So the idea that some part of our visual field, we may not be able to see it, but as soon as our eyes shift position, which they do many times per second, then what's going to happen is that area that was covered by the blind spot will now suddenly come into view. So it's not really a functional limitation to vision there because of that. So let's talk about the photoreceptors themselves. Here we have the rods and the cones, right? There are many differences between the rods and the cones. And what we're going to see is that they really kind of serve two separate visual systems. The rod system and the cone system are really kept separate, and the information that is encoded by the rods is different than the information encoded by the cones, and there's very little integration between those two systems. And what we'll see if you look at this kind of like point-by-point comparison here is that in many ways they are the opposites of each other. So for example, the rods are very sensitive to light, which means they can function in low light conditions, right? Because if they have a very high sensitivity to a weak light stimulus, that means that you can use the rods to see in the dark, right? Not, of course, in absolute darkness when there's an absence of light, but in the majority of cases, even in a dark night with all the lights in your house turned off, there's still some source of light somewhere, right? Maybe it's a small LED from some electronic device charging somewhere, but of course, starlight, moonlight coming in from the outside and street lights from the outside, maybe providing a small amount of light. And so you can still see. But that's using pretty much just rod vision, right? The cones have poor sensitivity. So that means the cones need a much higher intensity of light to be able to function. But by high intensity, I just mean sort of normal daily illumination. So sunlight, for example, is more than enough to be strong enough to stimulate the cones, but even also just a lamp in your room, right? As soon as you turn on a lamp at nighttime, you are stimulating cones at this point. And then, so it seems like the rods might be better than the cones because they have a better sensitivity, but now look at this next point and we see it's a trade-off, really. So while the rods have better sensitivity than the cones have better acuity than the rods. And so what's the difference here? Sensitivity is related to something that in perceptual research is called your absolute threshold, right? What's the smallest amount of stimulation that is needed to activate a receptor? So in hearing, for example, that would be what's the weakest sound that you can hear, right? And in vision, this would mean what's the faintest or weakest light that your eyes can detect, right? And so in this case, when we're dealing with those very faint, weak light stimuli, it's the rods that can detect these things and have a lower threshold compared to the cones, right? Acuity is your ability to detect small details, right? So once we're over threshold, we still have the idea that some stimuli are very large and obviously easily seen and noticeable, and others are very, very small. It's basically like when you're getting an eye test when you're getting a driver's license or just at the eye doctor, right? And they want to see what's the smallest level of detail that you can see without the need for correction, for example. And so that high acuity comes from the cones now. And if that seems like there might be a trade-off here, that gaining sensitivity as the rods do causes a drop in acuity, that is in fact true. But we need to explain why this is the case. How is it that we cannot seem to have both high sensitivity and high acuity at the same time. And it has a lot to do with exactly how the rods achieve this sensitivity. And this is through a concept we learned in the previous chapter called spatial summation, right? So let's think about spatial summation. Remember, in that topic, we addressed the idea first is temporal summation, right? That a series of nerve impulses all arriving at a neuron in rapid succession might sum together their activity. But in this case, we're talking about spatial summation. So what might be happening is to imagine a single postsynaptic neuron that's receiving input from numerous other neurons. And those numerous other neurons are all sending in a graded potential. And so that individual graded potential from each one of those individual neurons is too small to stimulate the action potential. But since they all arrive at the same time, they add themselves together to create a large enough signal to cross that postsynaptic neuron's firing threshold, and then you get the EPSP, right? Well, that's exactly what's happening with the rods here. The rods are using spatial summation. So that means that we could have a neuron that's in the retina, that's not a photoreceptor, it's just a regular neuron. And what it's doing is it's essentially monitoring the activity of several rods. Let's say it's 100 rods, right? So imagine a grouping of rods kind of organized in a circular pattern somewhere on the retina. And lying literally on top of all of those rods would be a single neuron somewhere that has dendrites that are spread out, and all of the axons of all of those many 100 rods are all synapsing onto that one neuron with its many dendrites all fanning outwards, right? So what we're going to say here is that this single neuron, I'm going to keep calling it a postsynaptic neuron, has a receptive field that includes all of these 100 rods. Now let's imagine we're in a situation where we are in a very, very dimly lit environment. You're in a dark room late at night, there's no lights on, maybe just a little bit of moonlight or starlight streaming in through the window, and you've been in this room for 30 or 40 minutes. So you're fully dark adapted, which means that your rods have fully adapted to this low light condition. The cones can't function. But this very low amount of light is now entering into your eye, and it's falling across your retina, right? So there's this very dim light distributed across your retina. And this causes a very weak activation of each one of these rods. And so each one of these individual rods that are part of that receptive field of this one postsynaptic neuron is sending a very weak graded potential to that postsynaptic neuron. Individually, each one of those graded potentials is too weak for the postsynaptic neuron to respond and to fire an action potential. But because we get to take all 100 of these rods and sum together their combined graded potentials, now the total input to that postsynaptic neuron is strong enough to trigger an action potential. And now that's going to send a signal back to the brain. And that postsynaptic neuron is essentially able to tell the brain, there's enough light at this part of the retina for you to see something, right? So there's light here, and now you're seeing, right? So that's how the brain will be able to detect the activity of those rods, because they respond in the low light, but they're summing the responses together so that the other parts of the brain can actually fire in response to the light. With the cones, we don't get the same kind of spatial summation. Let me exaggerate it here and say that it's really more one-to-one in terms of how the cones converge onto postsynaptic neurons. So if we have now that same very low amount of illumination distributed across the retina, again, the cones might respond to it, but the weak graded potential that the cone sends to its postsynaptic neuron is too weak for that postsynaptic neuron to respond. And since there's only one cone mapping onto one postsynaptic neuron, that postsynaptic neuron is not going to fire, right? It's not receiving summed graded potentials from multiple cones like the ones that are watching the rods are, right? So what we're really suggesting here is that there are some of these postsynaptic neurons in the retina that are only watching rods, and they are receiving a large combined spatially summed set of graded potentials from the rods that will help that system respond in low light conditions, whereas the cones and the cells that are monitoring all of the cones are monitoring far fewer numbers of cones. and therefore they are not able to get that same spatial summation effect, and they will not be responding in these low light conditions because those graded potentials are too weak to activate these particular neurons in the retina. Now, that helps explain why the rods can be sensitive to low light and the cones are not, but why does it trade off with acuity? And this is where we need to understand how information about shapes are encoded into neural patterns. And one thing that we need to remember is that shape is about detecting an edge from on the shape of an object, right? Imagine you're looking at a square. So that means you have to be able to detect the four edges of the square and distinguish the edges of the square from either the inside or the outside boundary of that object itself. And that involves what's referred to as contrast, which is technically a change in brightness. So if you're looking at a dark square on a white background, that boundary between dark to light is the contrast edge, right? And the way that is detected by the visual system is that a part of the retina is receiving that lower light reflecting from the black square, right? And then some neighboring part of the retina is now receiving the reflected white light from the white background outside of the square. And so you're going to have a differential pattern of illumination on the retina because some of it's dark and some of it's light from the pattern of dark and light on the square that you're seeing. So now let's imagine, for example, that when you're looking at this square that is dark and on one side and light on the other, that that edge is falling within the receptive field that I was talking about previously for that one postsynaptic neuron that's watching 100 rods. So if that boundary between the dark area and the light area falls within the receptive field of that particular neuron, all that's going to happen is that maybe half of the receptive field will be stimulated by the lighter area has a little bit more reflected light. And so let's say 50 neurons, 50 rods are being stimulated by the light, and the other 50 are not being stimulated because they're looking at the dark part of the visual field. Well, the activity of those 50 neurons, those 50 rods, I mean to say, might still be enough to send to sum their activity and create a strong signal to activate the postsynaptic cell. So now the postsynaptic cell is firing, but notice that postsynaptic cell can fire either if the entire 100 rods are being illuminated, or even if sometimes only 50 of those rods are being illuminated. If those 50 rods are receiving a strong enough input, then their summed graded potentials might still be strong enough to activate the postsynaptic cell. So the postsynaptic cell is firing, and its response doesn't seem to depend on whether or not all 100 rods are active, or maybe sometimes just 50 of the rods are active. And that means that whatever signal it sends to the brain is just the same. I'm firing, I'm telling you that there's light somewhere in my receptive field, but it doesn't seem to be able to provide any additional information about what kind of pattern of light that might be, and whether it's a uniform distribution of light or whether it's a non-uniform pattern that contains a edge or boundary between a dark area and a light area. So that means that this rod system is not going to be very good at detecting edges and boundaries between things, because it really is only going to provide information about very subtle general changes in brightness across the visual field. But the cones, on the other hand, because their alignment to those postsynaptic neurons is, as I said, more one-to-one in nature, they don't get that spatial summation, so they have to have bright light in order for the cones to send a strong signal. But in a bright light condition where the cones are sending a strong enough signal, now what might be happening is that if there is a differential distribution of light across the retina, so there are some areas that are light and some areas that are dark, especially like if you're reading a book and you're looking at printed letters on a page, black ink letters on a white page, for example, that's going to create a differential distribution of light and dark across the retina. Some cones will be stimulated by the whiteness of the page because that's more light coming off of that. Others will be less stimulated because they're looking at the darker part of the letters. And that differential activation of the cones is preserved as a differential activation of the postsynaptic neurons that are monitoring the cones. And so that differential pattern is maintained as when that signal is transmitted back to the brain, so that the brain understands, okay, yes, this is now a differential distribution of light, dark across the retina, and the cones can provide that detail, but the rods are not, right? So the rods are just giving us those overall variations in brightness levels, but the cones are giving us information more about the distribution of light across the retina from one cone to another, like that. So they really trade off, right? In order to get that acuity, you have to have more of a one-to-one mapping of individual cone to individual postsynaptic neuron. So you're not going to get any summation, right? But if we have bright enough light, we can see in detail. And of course, it makes sense from our experience that when you're in a low light condition, you don't see things in very clear detail, right? You can't see edges very clearly. Everything's a little blurry. But in a high light condition, you can see depth and detail very clearly. This also relates to color as well, because you might also notice that in the darkness, you really cannot see colors, right? If there's a colorful light on, like again, I mentioned, like you maybe have a phone on a charger, you might see a red light or green light or amber light or something on some LED for some charging device, you can see that. But other than that, the rest of your field of view is really going to show only shades of gray, right? And that's because in order to encode information about color, we need to understand the nature of how the rods and the cones work, and that there are 3 different kinds of cones, whereas there's only one kind of rod. And I have a graphic coming up that will explain how that works here in a second. Now, this last point here about the fovea versus the periphery, this also should now make some more sense to us, because I said earlier that the fovea is the area that when we're looking directly at something and we get that better detail. Well, that's because the fovea is full of cones, right? And so having only cones actually in the fovea, and they're all very densely packed in there together so that they can extract a lot of more detail from the any differential pattern of light falling across the fovea. That's where we're going to get that detail. We do, of course, still have a few cones located throughout the entire visual periphery, but most of them are densely packed in the fovea, whereas for the rods, there are no rods in the fovea. They're only found in the periphery. And yes, there are more rods than cones, but again, this is the idea that those rods are so spread out across the periphery that we have a lot of those rods so that we can see these variations in brightness levels and have a lot of them all summing together onto these postsynaptic neurons. Fewer cones, but again, there's enough of them still, 6 million per eye, that we have plenty of cones there to extract fine detail. Now, here is a graphic as well as a little electron scan microscopic picture of the rods and cones, so you can get an idea of why they're called rods and cones, which is potentially not very interesting, right? These cells are really, they have a portion of that cellular anatomy that can either be rod-shaped or cone-shaped, and we can kind of see that there. But what we need to understand about these cells is that, they are neurons, they are sensory neurons, but they don't look like the neurons that we've seen before, right? Where they don't have dendrites in particular. They have an axon, they have a cell body, but this outer segment that's labeled there on the drawing in the middle, we see that this has this little thing in there, kind of looks like a rattlesnake tail, right? But it's really just a stack of discs that contain chemicals called opsins, and rhodopsin is one of the main types of these chemicals. But these chemicals are technically pigments, right? And what do pigments do? Like in paint, a pigment absorbs certain wavelengths of light. And that's what's exactly going to happen here, is that the chemicals here in the outer segment of the rods and the cone absorb certain wavelengths of light. And when this happens, these chemicals break down into a variety of ions. And what's going to happen if we start to see a change in the distribution of ions inside the rods and cones is that that's going to change the membrane potential of the rods and the cones, which may trigger a signal that travels down the axon and synapses onto these various postsynaptic neurons that I've been talking about. And we see that on the left here, that on the retina, we see all of these rods and cones all lined up together on the retina. And what's happening is that we can see the little axons there. If you can see my mouse pointer, sometimes you can see it. With Zoom, it's hard to tell sometimes. All these little axons are branching off of the rods and the cones, and they're synapsing onto these little neurons that we see in blue here, right? And then these blue neurons are those postsynaptic neurons that I'm talking about that might be monitoring mini rods or just a few cones, right? And then in turn, those blue neurons are going to send an information to these other yellow neurons. And then we can see all of these yellow neurons have axons that are bundling together to form what will be the optic nerve. And we might be able to see that in this on the far left of the image here. We see the light ray coming into the eye, with the lens focusing it to the fovea there. And we can see the optic nerve leaving the eye there, right? So we need to maybe talk a little more about what these other neurons are. But before we do that, let me explain the color aspect of how the cones work. So color vision is achieved because we have three kinds of cones, at least for a person who is considered a trichromat and has normal color vision. People who are colorblind may only have two of these three kinds of cones, and they are considered a dichromat. And they still have some degree of color vision, but they don't have the ability to see the full range of colors that a trichromat can see. If a person only had one kind of cone, which is pretty rare, then they would be considered a monochromat, and they would only see in black and white, which also explains why the rod system doesn't code information about color, because there's only one kind of rod. So how does color vision work with the cones? Well, we can call it here, these three cones as S cones, M cones, and L cones. That stands for short wavelength, medium wavelength, and long wavelength cones. We can sometimes call them blue cones, green cones, and red cones, but that's tad misleading because they're not particularly, they don't have that color, but it's the idea that they kind of respond to light in that wavelength that roughly corresponds to that color, though not quite so true for the L cones there, that peak is closer into the yellow-orange part. But anyway, let's take a look at the L cones, for example. So we see that it's a curve, and this curve represents the sensitivity and therefore the response of the cone to a light of a given wavelength. So if the light is exactly 559 nanometers, the L cones are most sensitive to that, and they have their strongest response. And by strongest response, I'm talking about the graded potential it sends to these postsynaptic neurons. You get a strong signal going there, right? But it's not the only signal. So we can increase the wavelength up towards 600, 650, 700, and the neuron still responds, but obviously it's a weaker response. And likewise, if we decrease the wavelength down towards 500 or even around 460 or so, it kind of bottoms out, but it also kind of flips up there a little bit as we move even further down towards 400. And the green M cones, that is very much a similar looking curve. It's just shifted a little left here with a peak at 531 nanometers. And then the S cones take us down to 419 nanometers for their peak response. And then there's a sudden drop off there just below 400 because the eyes simply just don't respond to anything below that wavelength, right? And so when it comes to seeing a particular color, it's not really just about the activity of a single cone responding to that color. It's really about the combined ratio of these three systems. So for example, let's imagine we're looking at light that is 500 nanometers, which would be somewhere in the green band of the spectrum, right? What you would get there is you would get a moderately strong response from the M cones, right? Because that's partway down their curve, but you're also going to get a modest, perhaps medium-low response from the L cones, and then you're going to get a low response from the S cones. And so that unique ratio of medium M response, medium-low L response, and low S response, that particular ratio is going to now be interpretable by the brain as a green color. But if we go up towards, let's say, into that orange, around 625 nanometers, then we're going to get medium low from the L cone, low from the M cone, and maybe no response at all from the S cone. And that particular ratio equals seeing orange, right? So all of these different colors of the rainbow that the eye can detect can be encoded by some unique combination of the activation of the three different cone systems. And this also explains why the rods can't give us color, because there is no differential activation. Even though the rod has a peak response at 496, and it responds to a banding of wavelength there, it cannot encode that as color. Instead, it's only encoding it curiously as brightness levels, right? Because if the rod is more sensitive to light at 496, that means when you're in a dark environment and you're looking at something that might be green, you're not necessarily going to see that it's green, but you might notice that it's easier to see it at night. It will appear a little bit brighter because the rod gives a stronger response to the light reflected from that object. So the light reflecting from the green object in a low light environment is still going to be 496 nanometers. But that's just encoded by the eye as that seems to be something easy to see, so it's brighter. Conversely, if there's a red object in a dark room, it will look almost black because the rods simply are not responding to any light reflecting from that object. So you can try this experiment if you want, right? So set up something that is like blue, green, yellow, and red side by side that are all equivalent in terms of their size and brightness. And then just set them side by side in an open part of a desk, and then turn off the lights and wait half an hour until you get fully dark adapted. And then you might notice that the things that are bluish green are a little brighter in the dark compared to something that's orange and red. Now, I keep talking about all of these postsynaptic cells that are monitoring the photoreceptors, the rods and the cones, and having a so-called receptive field that includes several rods and a few cones, perhaps. What are they exactly? Well, here's a drawing that's going to help us visualize the organization of the retina a little bit better. So now at the bottom of this drawing, we have the photoreceptors, the rods and the cones, and we see their little axons spanning up to these dendrites of these other cells called bipolar cells. And those bipolar cells are sort of vertically oriented. So they're sending a signal further on up to the top to ganglion cells. And it is the ganglion cells that are monitoring all of this stuff. And then their axons are the ones that bundle together to form this optic nerve, which leaves the eye. So in that other drawing, the postsynaptic cells that were blue, those would be the bipolar cells, and the ones that were yellow, those would be the ganglion cells. But there's more going on here, because you might notice that there are some other cells that have more of a horizontal orientation. They seem to be taking input, for example, towards the bottom, they seem to be taking input from both bipolar cells and maybe even rods and cones. And the axon is traveling horizontally left to right across the retina. And then above the bipolar cells is another horizontally oriented cell that seems to have axons and dendrites that are spanning left to right. So these are the horizontal cells and also amacrine cells, right? So the horizontal cells are the ones that work to send lateral information sideways between the bipolar cells. The amacrine cells are the ones further up that send the signal sideways across the ganglion cells. What is this crosstalk? It's well, largely the horizontal cells and amacrine cells, their signal is largely inhibitory in nature. So remember the difference between an IPSP and an EPSP from before. So that's what the horizontal cells and amacrine cells are doing. They're sending inhibitory signals. But it's lateral. So this is often called lateral inhibition. And the way this works is, remember when we were talking about edge detection, right? You're looking at a square and you have to detect the contrast between the dark side of a square and the light side of a square. And that's an edge. And that means that then the boundary between an area of the retina that's activated because it's seeing the light side of the square and another area of the retina that's deactivated because it's seeing only the dark part of the square, we want to highlight that boundary. And one way to highlight that boundary is, let's just say that when we're looking on the left side, let's look at that one horizontal cell at the bottom that's monitoring some rods and cones there. And those rods and cones, because if they're stimulated by some area of light, that they will send an excitatory signal to their partner bipolar cells, which will then, of course, transmit that excitatory signal on up to the ganglion cells. But meanwhile, that horizontal cell, or what it's going to do is it's going to take that excitatory signal and it's going to convert it to an inhibitory signal. So it's being excited by the rods and cones, but meanwhile, the axon that it sends sideways is going to be something that inhibits the bipolar cells over to the left or to the right of this region of the retina, right? So this helps to highlight contrast, because if you have a certain area that's illuminated with light and an area next to it that is in the dark, by actively inhibiting the dark region, you're going to magnify the difference in the response of the light compared to dark sides of that edge, right? So we excite one area of the retina while we simultaneously inhibit a neighboring region of the retina. and that helps to magnify certain kinds of edges that would be there, right? So that's the goal here, is that all of that lateral inhibition happening between the bipolar cells and ganglion cells is part of a process of finding where are the edges of objects in our visual field so that I can start to map out the shapes of things, right? So the very earliest form of visual processing of information here, where are the edges so I can recognize shapes and patterns? That happens even within the retina itself, not even in the brain. Before we ever get to the brain, the retinal cells here are processing all of this information about the differential distribution of light across the retina. We also want to point out, to wrap this up here, that there are many different types of ganglion cells that do different jobs. As I've already mentioned, the rod system and the cone system work pretty much separately from each other. And so that means there's a bunch of ganglion cells that are monitoring only the rods. And these are the magnocellular ganglion cells. Magno means large here. So this is a large system because there are lots of rods, right? And of course, the information that it processes is consistent with what we know about the rods, right? That it works in low light condition. It processes information about variations in brightness and movement even as well, which we'll explain some more about how movement is processed a little later. The parvo means small. So in this case, the parvocellular neurons are the ones that are monitoring the cones. And of course, all of the information about fine detail and color, that's being processed through the parvocellular pathway. So what we'll see is that the optic nerve, as it leaves the eye and goes back to the brain, maintains the magnocellular and parvocellular paths, right? So there's a part of the optic nerve which contains axons that are all dedicated to the magnocellular ganglion cells. And another part of the optic nerve is only about the parvocellular ganglion cells and the cone signals, right? As it turns out, there's more, right? So we also have the bistratified ganglion cells. And this is part of what is referred to as the coniocellular pathway. If I remember correctly, conio means dust, and this means that these are tiny little ganglion cells that send a signal to the LGN, which we're going to talk about coming up here. It's part of the thalamus, and actually the magno and parvocellular pathways also go to the LGN. But the role that these ganglion cells play in conscious visual perception is not well understood. It might play some role in things related to other aspects of brain functioning that depend on light perception. And speaking of other aspects of brain functioning that depend on light perception, there's another type of ganglion cell at the bottom here, photosensitive ganglion cells. So what in the world? These are ganglion cells that actually contain the pigment melanopsin, and that means they can directly respond to light. But in this case, they are not part of any conscious visual perception of an object, right? Instead, what's happening is that these particular ganglion cells send a signal to a nucleus in the brain. So remember, a nucleus in the brain is not a cell nucleus. It is a cluster of neurons that are working together to perform a specific function as a grouping, right? And so this is a cluster of neurons that is technically within the hypothalamus, and the hypothalamus contains several such nuclei that do different jobs for different kinds of rhythmic and motivated behaviors. This is the circadian rhythm circuit. And in particular, it regulates our waking and sleep cycle, which of course needs to be regulated by the daily sunrise and sunset cycle. So we need, of course, visual input to this sleep circuit so that the brain, in particular the hypothalamus, knows when the sun is up and when it's daytime, and that changes the rate of hormone production related to arousal, right? And then at nighttime, the suprachiasmatic nucleus knows that it's dark. And of course, then that starts to trigger melatonin production, right? Of course, melatonin is the hormone / neurotransmitter that's involved in making us feel sleepy, right? So this is all an important part of this. Now, you might wonder, what in the world is this word suprachiasmatic? And as we get further into these notes, we're going to learn what the optic chiasm is, and that's going to then from that we'll understand what suprachiasmatic means, because it means that it's supra here means sitting just above the chiasm, the optic chiasm, which I have listed here on this next slide as the junction of the optic nerves. But this is a good part for us to stop at this point before we trace that signal any further back to the brain and understand what's happening deeper in the brain. I think we've covered enough here just by talking about the activity of the retina and that'll be that.